Okay, so experiment 17 is about the effects of environmental conditions on growth and survival of microorganisms. And it is a long one, so let's get started. So the general overview is that we're going to be looking at different environmental conditions like temperature, pH, UV radiation, osmotic pressure, and see how they affect the growth of the bacteria. And what this tells us is that there's different ways that you classify bacteria based off of their effects and how they are basically the effects of these certain conditions on the bacteria. And we're going to be looking at that and major trends that go along with these different conditions. So part A looks at the effects of temperature on bacterial survival. So what the general trend is that you have some bacteria live in cold temperatures, others live in average temperatures, and some hotter. So they can live at a huge range of as a whole, bacteria can live in a huge range of temperatures. Now, each specific bacteria has a very narrow width of temperatures they can live at. They do have a range of growth. It's not just like one temperature because, environmental conditions vary vastly. And so if you like hotter temperatures like volcanoes and stuff like that, you're what's called a thermophile. And actually, if you live in a volcano, you're an extreme thermophile, right? But if you like hotter temperatures, you're a thermophile. If you're a mesophile, you like average temperatures. And if you're psychrophilic, then you like colder temperatures. And what we can do is that if the temperature that you have exceeds the range of temperatures, you start to denature proteins and nucleic acids and stuff like that, and you can effectively destroy the cell. And so pasteurization kind of takes advantage of this property in that they kill things. So if you're pasteurizing milk, for example, you heat up the milk to where you kill the bacteria that are in it, but not affecting the milk, and then you can have a longer shelf life for the milk. And then a fridge and a freezer, or an ice box if you call it that, what they do is they make the temperature colder, and then it ceases enzymatic activity, not allowing it to grow as effectively. So bacteria and microorganisms still grow in places like an ice box or fridge or something like that, but it's seriously reduced compared to if you just leave it out on the counter, right? And so what we did in part A was that each person had a bacteria and a temperature. And you have this chart right here that shows you where you sit, gives you the type of bacteria and the temperature. And what we did was these three bacteria right here were tested for their exposures to various temperatures. So the important thing to note here is that it's not their ability to grow at these temperatures, it's their ability to survive these temperatures. And we'll see that within the experiment. So what we did was we took 10 microliters of 100 microliters of bacteria culture and put it in nutrient broth. And we took from that nutrient broth and streaked it onto a plate. So we divided the plate in 4 ways, and we labeled 1, 0, 3, 6, and 9. And we streaked the first one into the 0 quadrant. And then we took this bacteria culture and then we put it in our assigned temperature, right? And so what that does is that it exposes the bacteria to the temperature, but it's not measuring the growth. And so if the bacteria will affect it, so if you put it at 100 degrees Celsius, for example, and if the bacteria dies at 100 degrees Celsius, then you're going to have dead bacteria. So when you go in 3 minutes later to streak it on your 3-minute quadrant, then you're not going to find going to find seriously reduced growth in that 3-minute quadrant. And so that's exactly what you do, is that you just stick your tube into the temperature, and then you streak it into the quadrant that is at the amount of time after you put it in there initially. So you put it in there for 0, so that was your first one, and you put it in there for 3, then you take it out and you streak it, and you put it in there for 3 more minutes and you take it out and streak it, and then you put it in there for 3 more minutes and you take it out and streak it, and then you get 9 minutes. So it's a total of 9 minutes, but you want to streak after each time period. So what's important to note here is that E. coli die at around 60 to 100 degrees Celsius, And then Bacillus megaterium have endospores, so they can actually survive these higher temperatures like 100 degrees Celsius. So those are two important notes there. But again, the really important thing to take away is that we don't measure the growth per se. We measure the survival here. All right, now part B is the effects of pH on microbial growth. So we did that one a little bit differently. What we did there is that, well, okay. So first of all, just like with temperatures, every organism has an optimal pH that they grow at. So you have a range. Usually it's plus or minus 1 pH unit, but again, that even strays from bacteria to bacteria. But the way to classify these are acidophiles, like acids, right? Neutrophiles, like neutral conditions, like pH 7, and then alkanophiles, like alkaline conditions. Basic conditions is a synonym, and so they like the higher ranges on the pH scale. And what y'all did was, as a bench, y'all tested the ability to grow at these pHs. So what y'all did was that each group of 4 had 3 sets of 5 different pHs. So you had 1, 2, 3, 4, 5. You had pH 3, pH 5, pH 7, pH 9, pH 11. So these are very acidic pHs, and these are very basic pHs. So you inoculated bacteria in one set of 5. So you took a bacteria and you have 3 tubes of pH 3, 3 tubes of pH 5, 3 tubes of pH 7, 3 tubes of pH 9, 3 tubes of pH 11. And you took one of these tubes of pH 3 and put one bacteria in them. So E. coli, for example. Then you took another one and then you took the pH 5 and put E. coli in it. Then same thing to 7, 9, 11. And then you did Saccharomyces cerevisiae, which is yeast, and you put one of those. So you put a sample of that into 3, sample that into 5, sample that into 7. So you use each of your 3 sets. And the thing to note here is that yeast, these Saccharomyces cerevisiae, grow at very low pHs, kind of, so between 4 and 6. And then the alcogens grow from 9 to 11, so they really like the more basic range. And so that was part B. Now, part C was the effects of UV radiation. So UV light can be used to sterilize surfaces. It doesn't really penetrate very well, so it can't really go through things, and we'll see that in our experiment. But the way that it sterilizes things is that it forms these things called pyrimidine dimers. In your lab manual it says thymine dimers because thymine is a type of pyrimidine, but pyrimidine dimers form because of these because of this UV light. And so what happens is that you have these dimers that are hydrogen bound to, if it's T right here, they're hydrogen bound to that A, right? But what happens is with UV light, it comes in and connects these two together instead of binding to the A, and so you get something that looks like this instead of these hydrogen bonds forming right here. And that seriously can affect the ability to grow, as we'll see. So what you did was you made a lawn of bacteria. So you took a plate and you just streaked all over it. And then we took an index card and we blocked one side of it, and then we left the other side exposed to UV. And so there were 3 different bacteria. So there was E. coli, Bacillus megaterium, and then Deinococcus radiolarians. And what we found was that some of them, so the UV light couldn't penetrate past this card because it has poor penetrating ability, and so it didn't actually get to those bacteria, while it did get to the ones that were just open and exposed. And so the big results here is that Bacillus megaterium, what they do is they repair DNA to survive it. So the amount of mutations that are forming right here are almost equal to the amount of repairs that form right here. And so UV light does not affect the growth as much as it would something as our control like E. coli. And Deinococcus radiodurans has pigmentation. So what you'll learn is that things that have pigmentation have, if you're taking organic chemistry, you have a highly conjugated system. And what UV light does is it affects the conjugated system instead of affecting other things like proteins and things like that. And so the pigmentation actually kind of takes on the UV light instead of the proteins and the nucleic acids. And so the pigmentation is taking it on instead of these thymine dimers, and so the pigmentation is really important into radiodurans, like having a good durability to the radio waves. Just kind of a way to remember it, I guess. So radiodurans is the ability to survive UV radiation. Okay, so the last part was part D, the effects of osmotic pressure. So there are a lot of terms in your lab manual that are very similar to the terms you learned in 1201, but they're very important, so you want to make sure to revisit those. I know I missed one when I took the test, and it made me mad because I could have gotten a better grade. So make sure to go over all those terms like hypertonic, isotonic, hypotonic, and then you'll see terms like halophiles, extreme halophiles, halotolerant. And so just make sure you know all the vocab for this lab. That's very important. But what we did for part D was that each person at the bench had a specific salt concentration on an auger plate. So you had 0.5 degrees NaCl, 5% NaCl, 10% or 25% NaCl. And so what we did was we divided into 4, and then we streaked a different bacteria in each column, in each quadrant. And so then we let them inoculate, and then we assessed the plate for growth. And then as a group, we kind of compared the different growth rates at the different concentrations of salt. And so the concentrations of salt are directly correlated with the concentration, with the osmotic pressure. And so if bacteria had osmosis that put water from the inside of the cell to establish an equilibrium with the higher salt concentration on the outside of the cell, then the cells would desiccate and then die out because they can't grow as well because they don't have enough water. And so that's the important part from part D. So the take-home from all this is pretty much a lot of stuff and how experiments work in a microbiology lab. And so it allows you to see why the autoclaves work, because what the autoclave does is it does a very high temperature and very high pressure. And so if you can overcome even the highest temperature and pressure that of all the bacteria that we're using, like if you do it at a high pressure at a high temperature for 15 minutes, then you kill all the bacteria that even form endospores. And so that's an important property that we take advantage of here. So we also see how sterilization with UV works, right? And so if you use UV light, you can kill all the things that are on a surface, and by mutating all the things, and so it doesn't grow, and so you actually sterilize the area like that. And so now we know how to expand our abilities to classify. I know it can get a lot of confusing stuff, and now we have to learn all these like different vocab words, but you'll do need to know it for the written portion. And know that you can mix and match all these classifications too. And so you can have something that is halotolerant and extreme thermophile or something like that. Is that each one has its own classification, but within that classification it also can expand on all these other ones. So these experiments are not isolated things in the world, and each environment has a specific temperature, pH, UV exposure, and osmotic pressure. And so that's what the true take-home is that each bacteria has to deal with these things and other factors as well. These aren't all of them. And so it's about seeing how the bacteria interact with the environment and the natural world around us, and then what maybe we can take advantage of and not take advantage of.